Should start. So I'm confident we have all heard about the Black Death, referring to the plagues of the Middle Ages. We have probably also heard about the Emperor of all maladies, referring to cancer. Kafka nicknamed tuberculosis the germ of, let's see, right, the germ of death itself. So today we're gonna hear another story, a story told by Mikkel from D-IAS Assistant Professor. A story that is called, I guess, the cloak of invisibility, the story about the captain of the men of death. Yes, the story of the bacteria Streptococcus pneumonia. And we're all, of course, very excited to hear that story. So Mikkel started out as an undergraduate student here at SDU before moving with Professor Ken Gerdes to Newcastle University, where Mikkel then eventually got his PhD. And then Mikkel returned for a few years here to SDU to do his postdoc. Did a second postdoc in, let me pronounce this right, Jan Willem Wiening's group in the Netherlands, in Groningen. And again, luckily for us, Mikkel then once again decided to return to Odense, where he then since 2016, last year, has been heading his own research group. And they are now studying this very fascinating bacteria, Streptococcus pneumonia, which has the ability to change lifestyles in order to, as I understand it, infect human beings, many of them actually children, and be the cause of such devastating diseases as sepsis, meningitis, and also pneumonia. So I guess with that, I'll give the word to you, Mikkel, and we're very excited to hear a story.
Take it away.
Yeah. Yeah, great, thank you.
Thank you for the introduction. And I guess thank you for the invitation as well. I've really looked forward to this day, also because I get to talk about my favorite topic, which is the Streptococcus pneumoniae. So today I was a bit unsure of what I wanted to talk about. It should be something exciting and something in this room, something curious. And I realized, and I think this is how most scientists actually operate. The experiments that you are conducting right now, they are the most exciting ones. So this is what I'll talk about today. The projects that we have in the lab that are ongoing right now. So they are not finished, none of them. And even though it seems that it's different projects, I think they very much tell the exact same story. And I think you'll agree with me when we're done for today. The cloak of invisibility, I think the title will become evident to everyone in a couple of slides' time. So, but as I said, all the projects are telling the same story. And I think for today, our story will begin over 100 years ago with this guy, Sir William Osler. He was a Canadian physician, and he was the guy who actually coined the term the captain of the men of death. So he was also studying this bacteria, Streptococcus pneumoniae, and in 1904 he said that in the mortality bills, surely the pneumococcus has become the captain of the men of death. So this was in 1904. In 1904, Streptococcus pneumoniae was the most widespread and fatal of all of acute infectious diseases. We've had a century of medical advances, the development of vaccines and antibiotics. But by and large, this statement is as true today as it was then. So let's have a look at the bug itself, the Streptococcus, the pneumococcus in short. It looks like this. It grows in these chains, like pearls on a string. They only have one natural habitat, and that's humans. So they are pathogens. They enter humans through either the nose or the mouth, and then they colonize the nasopharynx, which is, maybe you see better if I move over here, colonize the nasopharynx, which is this back part of the nose. And from that colonization site, it can then move and cause some less severe diseases like common middle ear infection or infection in the sinuses. I think we've all had that. I know that it's not really such a big deal. It's not really fatal. It's an opportunistic pathogen. It means that a lot of people are carrying this bacterium asymptomatically, and they will never become diseased in any way. They are just carrier of the bacteria. And they carry them here in the nasopharynx, and from that place it can then colonize other sites of the human body and cause what we term the invasive diseases. And these are much more severe. And these are in particular pneumonia itself, which is infection in the lung. This is what we most typically see with this bacteria, hence the name. It can also travel up and cross the blood-brain barrier into the cerebral spinal fluid, causing meningitis, or it can just cause a persistent infection in the bloodstream bacteremia. Needless to say that if left untreated, most of these diseases are often fatal. And the bacteria itself, for that to really be virulent, that is the ability to cause disease in its host, they have this capsule structure surrounding the individual cells. The capsule is a thick layer of polysaccharides composed mostly of just simple repeats of sugars. And the capsules surround the bacteria like you see in this image here. But before we go on and talk much more about this capsule, I'll just talk a little bit about the persons who are actually at risk of catching some of these horrible invasive diseases. And in particular, it's old people and it's children. Children 5 years of age or younger. They are the particular risk groups of catching these diseases. There's also a geographical component to this, where we see that these invasive diseases are much more predominant in resource-poor countries in Asia and in Africa. But if we just look at the children alone on a worldwide basis, pneumococcus itself is responsible for the death of more than 1 million children every year. This is really quite a significant number. It's 11% of the total deaths in that age group alone. And these are numbers that are excluding HIV and AIDS patients. And I think, as you know, most of these patients, they end up succumbing to common pneumonia as well. So these are not included in these numbers. It's just pure disease in healthy beings. It's not, I guess I'm really far away from the computer. It's also the children who are carrier of the bacterium itself. In particular, that's those small children of 5 years or younger. It turns out that a kindergarten is the perfect incubator for streptococci, where in kindergartens, this is also true in Scandinavian countries, where the carriage rate can be up to 100% in kindergartens. So the kids will share toys as they would share their streptococci as well. And the different bacterial strains, they will circulate within the kindergarten, and the children will bring it home to their parents, to the adults. When we then look at the carriage rates in adults, it's much more diverse, but if you're a parent to young children, you're much more likely also to be a carrier of streptococcus. It's not with these diseases that we are completely helpless to do anything. As I said, we've had a century of great discoveries, particularly in antibiotics, of course, and there is also an effective vaccine against streptococci. There's just two things to note. I won't really talk about too much about it, but the World Health Organization, the streptococci, as with most other infective bacteria, they are now becoming increasingly resistant to antibiotics. So the resistance of the pneumococcus now has a worldwide distribution, which is of great concern according to the World Health Organization. But of course we do have the vaccines, right? We do have effective vaccines. With vaccines, there's just another concern to consider. And that's something called serotype displacement. So currently there are 98 different serotypes of streptococcus. A serotype basically means that the capsule comes in 98 different flavors. There are 98 different chemical entities making up the capsule structure. The vaccine is called PCV13. It was introduced as part of the Danish National Vaccine Program in 2007. The number you should look for is 13. It means that the vaccine is currently covering 13 of the 98 different serotypes of streptococci. And what serotype displacement means is simply that, yes, we do have a vaccine and it is very effective. So when we vaccinate at this point, the serotypes covered in the vaccine, they of course rapidly decline, but that just leaves room for the types that are not covered in the vaccine, and they will just basically take its place instead. And as you can see, the overall rate of carriage, that doesn't really change. I really wanted to include this slide as well, I think. We've known for a long time, I said that the capsule is a major important determinant of the streptococci being able to infect the host. And we've known that since the Second World War. And this guy, Oswald Avery, I think he did one of science's most famous experiments. So he was working with streptococcus as well. And what he had was, he had some live cells that were non-virulent, so they are not capable of causing disease. And he mixed those with a strain, a virulent strain, so it is capable of causing disease, but heat killed those cells. He mixed them in a 1-to-1 ratio. He split them in 2 tubes, and in one tube he removed the proteins, in the other tube he removed DNA. I'll just remind you that the flow of the genetic information, of course, is that the DNA, the blueprint of the cell, it doesn't really do anything. It needs to be copied into RNA, and that RNA is translated into the functional workhorse of the cell, the proteins. So what he did was he removed the DNA in one tube and the proteins in the other tube, and then he fed those tubes to mice. What he saw was that when he removed proteins, the mouse died, but if you removed DNA, the mouse lived. So by this experiment, he concluded that somehow the virulent strain is capable of transferring some of its infectious traits to the non-virulent strain. And it was the DNA that did it. So by this experiment, he proved to everyone's satisfaction that it is the DNA that is the carrier of genetic information, and not proteins, which was actually a common belief at the time. This was in 1944, and what Avery didn't know at the time, but what we know now, is that the reason the mouse all of a sudden died from the non-virulent strain, what was transferred from the virulent to the non-virulent strain, were the genes that are responsible for producing that capsule polysaccharide structure. So from this experiment, we also know today that the capsule is actually the most critical part of the pneumococcus during that infection cycle. Without it, the strains are simply not capable of causing any disease. So you'll see that exactly why Avery's mouse was dying. We'll see that in a minute. For now, this is to help you, just to walk you through the next couple of experiments. This is something we do a lot, is that we tag the different cells with fluorescent proteins. I hope there are no red-green color lines, actually. If so, I'm sorry. What we do have is that we can label the cells that are capable, that produce a capsule with a green fluorescent protein, so they are green. You will see the capsule in this image as this black halo around the individual cells. And what we also do is we can then make a mutant, so it is a strain that is not capable of producing a capsule, and we will label that with a red fluorescent protein. And simply by just looking at the colors, we will know exactly if that particular bacterium is producing a capsule or not. So what you can do, and I think this illustrates very nicely exactly why Avery's mouse was dying. This is on a microscope slide, so you can take the two strains, the red and green capsule or unencapsulated, and mix them on a microscope slide, and then you would add in human macrophage cells. So the macrophages are immune cells. They are really the policemen of the human body. They are on a seek and destroy mission where would hunt and look for foreign bacteria, and when they encounter them, they will just eat them, kill them. So, and when we mix them like that, whoops. Okay, here we go. So have a look at these macrophages. As soon as they encounter the unencapsulated cells, the red ones, maybe this one, you can see that the macrophages very efficiently recognizes the unencapsulated cells. But if you notice the green ones, the encapsulated ones, they remain completely untouched. So the capsule, and the reason why Avery's mouse was dying, is because that the capsule really enables the bacteria to become invisible to the immune system. Of course, in this simple cartoon here, this is when the cells are inside the bloodstream. But for the cells to actually enter the bloodstream itself, they first need to colonize, right? They first need to enter either through the nose or through the mouth and start infecting the lungs. To do that, they first need to adhere and start to colonize on the surface of the human cell layer. And of course, as molecular biologists, this is the exciting part, right? Because this is where all the cell signaling takes place, telling the bacterium that it is now in the right place. This is the eagle is landing moment, right? And when we look at that situation, and again we have the red and green encapsulated and unencapsulated cells, and we will mix them. So in our experiments, we don't really, for ethical reasons, we don't do human experiments. We simply just grow the human cells. These are the blue cells I've tried to illustrate here in wells, and we can then mix the two different strain types in that well, and we will let them colonize. So that is binding to the surface of the host cell, like that. We can then wash away all the ones that didn't bind, and then simply look at what does it look like, who is actually colonizing that surface. And what you see is, if you're not red-green colorblind, is that it is only the red cells that are capable of adhering to the host cells. It's only the unencapsulated cells that can actually bind to the surface of the host cell. And the reason for this is because the bacterium has some adhesins that need to be exposed in order actually to make that contact and make that interaction. Some receptors that needs to be exposed to bind properly and tight. That big capsule is preventing that by simply being a bulky mask that masks these adhesins from ever coming into contact with the host cell layer. So this was the moment where the streptococci are coming in for the first time and adhering to the cell layer, colonizing the surface. But there's also the case of the infection, that moment that comes after this moment, right, where the cells are entering and penetrating, infecting the human host cell layer. That's another case entirely. So what we do the same thing again. We have different wells with human cells at the bottom. We can then, in two different wells, add the encapsulated or the unencapsulated ones. We will then remove and kill all these that are sitting here on top outside, and we can then simply count how many bacteria are now present inside the human cells. And when we do that, you will see exactly the opposite as you just saw for colonization. In this case, it is only the encapsulated cells that you find infecting the human host cells. This slide was just for you to appreciate the dynamics of this capsule structure. So as the cells enter, as they enter either the lungs or whenever, if they are even in the bloodstream and wanting to penetrate the blood-brain barrier, they come as encapsulated. But the encapsulated cells, they can't really sit on top and adhere to their host cells. For this to occur, it is only the unencapsulated cells that is doing that. And then, during infection, they need to turn on the capsule production again rapidly, because it is only the encapsulated ones that are capable of doing this feat. Then there is another process that I didn't really talk about. It's called transcytosis. It's basically where the bacteria just shuttle through the human cells and enter the bloodstream directly. This process requires the cells to be completely capsule-free as well. And as soon as they enter the bloodstream, they need to build a capsule again rapidly, otherwise they will be just simply killed by the immune cells. I'd like to come back to this, this initial attachment. As I said, this is the exciting part, right? This is where all the cell signaling takes place. What a lot of people has been doing, and what we're doing as well, everyone finds this the most exciting part, right? And what a lot of labs all over the world has been looking at is, so which genes are actually induced and which is turned off? Which genes are turned on and which is turned off? Okay, just remember that when the genes don't really do anything. So when I say a gene is turned on, what we are really looking at is the level of the RNA, whether it is going up or down. So, and when the cells then attach and adhere to that human host cell, there are two things of major importance. Well, there's a lot of important things, and a lot of exciting things going on, but there are two things we should talk about today. And that is, you'll notice that the genes responsible for producing the capsule, their expression is decreasing. I think that's not really as a surprise, is it? You just saw that it is absolutely critical for the cell to bind to the host cell that they need to have capsule removed. So it makes perfect sense that the genes are, that the gene expression is declining. What is perhaps a surprise is that genes for competence, they are massively induced. And competence in the bacterial world has nothing to do with how we are competent. In bacteria, competent simply means the ability to take up foreign DNA. So it's a process that enables for the bacteria to take up DNA from its surroundings. And it's a cascade reaction. So here's an outline of a cell, a bacterial streptococcal cell, and it's a response that is initiated when the cell senses a stimuli. In this case, for competence, it's called CSP, or competent stimulating peptide. The cell senses this signal, and then it relays this signal through the cell, and that involves a lot of different genes that are turned off at this point. Some are the genes that are encoding the proteins that are actually required to take up the DNA from the surrounding itself. But there's another thing happening at this point. That is that when this particular cell senses CSP from its surrounding, it will start to produce CSP itself, and it will transport it outside the cell. Now this would lead to a further stimulation of this cell to produce even more CSP. So this is a positive feed-forward loop that would just lead to the stimulation and production of even more and more and more CSP. This is exciting. We'll come back to that in just a minute. For now, I just want you to draw your attention to this protein. It's called LID-A. I think LID-A, it may be one of nature's most exciting proteins. It may not look like it, but it certainly is. So when LID-A is induced, it travels to the cell wall, and then it will start to degrade the cell wall. Remember that, okay, I didn't tell you yet, but the capsule is not the cell wall. The capsule is sitting on top of this part, right? So when LID-A starts to degrade the cell wall, it's like wearing a t-shirt. And if you're wearing a sweater at the same time and you take off your t-shirt, you also end up taking off your sweater, right? You just saw that competence is induced when the cells are touching the human host cells. So of course we were wondering if this is not necessarily a process because the cells want to take up DNA from the surroundings. No, rather it's a process that enables the bacterium to shed their capsule. This important part. So the way we do these experiments is that we, in a test tube, we will have the bacteria, and then we can add that competent stimulating peptide. We wait a little bit, then we spin down the cells to the bottom. So if competence is inducing the cells to shed their capsule, we will find the capsule material up in this liquid phase. So it's a matter of taking that liquid and ask, do we see any capsule? So running these capsule materials on gel is rarely a thing of beauty, but I think the result is quite clear nonetheless. So the presence of capsule will show up as this black smeary thing when you run it on a gel and visualize it. And you see it only happens in the cells that has been stimulated for competence. Cells that are not competent, they will not get rid of their capsule. I just mentioned that about the positive feed-forward thing. And I think almost per definition as a geneticist, these are interesting. And the reason for this is that positive feed-forward loops, like the one you just saw, they have the ability to create a bistable population. A bistable population, so I tried to illustrate it in this imaginary graph here. And bistability basically means that some cells in the population are expressing a set of genes that the other cells do not. It's a reasonably new thing in bacteria with having these bistable, well, it's not new for the bacteria, it's new for us. Reasonably new. And the cells, of course, they grow in completely identical media in the same environment. And yet, these positive feed-forward loops are capable of driving this bistability, creating these two different subpopulations of cells. And of course, so we immediately asked, is it so that induction of competence is giving that phenomenon, bistability? And the way we did this experiment is that we labeled all the cells with a green fluorescent proteins. So here, all the cells are green fluorescent. Then we took a gene that is only induced in competent cells and added and fused that with the green fluorescent protein. So now we can see all the competent cells, they become green, and we can visualize all the cells in one go because all of them are red. And asking, of course, do all the cells in our population become competent? And the answer, when you look at the overlay, is no. It's about half of them only, actually. I think I'll just pause here so we can consider the consequences of this, because it's easy to lose track of why this is so exciting. But it is. So remember that colonization, right? That initial attachment and adherence to a human host cell in the lungs or nose or where it is. The cells need to get rid of their capsule. And they do this by inducing competence. But also, what you just saw earlier is that the capsule-free cells, they are the ones that will light up as a beacon for the immune system. So, as a bacterium, you don't really want to just get rid of your capsule. And maybe you don't even want the entire population to get rid of its capsule. It's like committing collective suicide. I think for the bacterial purpose, as a bacterial population, from that point of view, this is a very clever strategy. Should we start, colonize and infect, expose ourselves to the immune system? Yeah, let's do half. And this is exactly what these cells are doing. At least, this is what we think they're doing. Because right now, these competent cells, so we know that competence is causing the cells to get rid of their capsule. What we don't know right now is that whether it is the competent cells, these ones, are these also the ones that are shedding their capsule? Of course, we believe that very much to be the case. That would be the logical thing, and this is exactly what we are pursuing right now. Now I'll just pause here and leave these competent cells alone and move on. You saw earlier that during this adherence to their host cells, to the human host cells, competence was induced. I think at least we now know why this is happening. They induce it so they can get rid of their capsule. But at the same time, you also see that the expression of the genes responsible for capsule production is turned off. I also think that makes a lot of sense why this is the case. We can't have that they are losing the capsule, and at the same time they are just producing all the proteins required for the production. So it makes sense that capsule expression needs to be turned off. But what we don't know is the proteins that are regulating this. How is this regulated? Or which genes are responsible for controlling capsule production? We wanted to look into that. And for this we made an unbiased genetic screen where we looked for capsule regulators. So we used transposons, which are movable genetic elements. For today, we don't really need to go into details with exactly what transposons are and what is happening. Just note that transposons are discrete DNA segments that has the ability to integrate themselves at random positions on the chromosome. So one transposon would go in, if it inserts itself in a functional gene already, you would disrupt that particular gene, creating a single gene knockout. We can then do that thousands of times and creating a library each consisting of different mutants with single gene knockouts, and some of them, just a few, they would have a gene knockout in a gene that is responsible for capsule production. So that cell would not produce a capsule, then it's just a matter of finding and isolating that cell and ask, where did the transposon go? Okay, I said it's a simple matter of finding these cells. It is very much looking for a needle in a haystack. So we needed to come up with a way where we could identify the needle, so to speak. And to do that, we created a reporter strain. Again, we took advantage of the green fluorescent proteins. So here are the genes that are responsible for capsule production. We took one of the genes and fused with GFP. So in our case, that means that a capsule producing cell would be green fluorescent. And in our library, there would be mutants that doesn't produce capsule. These non-capsule expressing cells would be non-fluorescent. So we just have a random pool of mutants where some of them, and these are the ones that we want to find, they are the non-fluorescent cells. And this is what we did. We created 2 libraries with more than 20,000 mutants in each. And when we look at the reporter strain, the wild type strain, the ones that are expressing a capsule, you will see that they are all green, they are all fluorescing. They should. It's not that the entire cell is green fluorescent, it's just that they are fluorescent at mid-cell. This is because all the capsule producing genes, they cluster like this at mid-cell and then produce the capsule from the center and out toward the cell poles. So, but they are all green. In our library, what we are looking for, of course, are the ones that are non-fluorescent. These are the interesting ones, like this one. So what we do is use fluorescence activated cell sorting, or FACS. It's basically just a machine that takes all the green cells, put them in one tube, and all the non-fluorescent cells and put them in the other tube. So all the green cells, we throw them out, right? We are not interested in those. We're interested in the ones that are non-fluorescent. So we can isolate those, and this is exactly what we get. A complete isolation of cells that are non-fluorescent. And then it's just a matter of asking, so where did all the transposon go in those cells? And this is what it looks like. Here are the 2 different libraries. Streptococcus has a circular chromosome, And basically each spike in this diagram, the higher it is, the more mutants we have isolated where a transposon is sitting at this particular location. You'll notice immediately one thing, and that is there are a lot of transposon hitting in a particular area on the chromosome. And when we take a closer look at this area, we see that it is all the capsule biosynthesizing genes themselves. And of course this was great news. This was fantastic because remember the screen was asking for the genes that are responsible for producing a capsule. So finding the proteins that are indeed building the capsule itself, this is what we wanted to see, right? But they're not really the interesting ones. The interesting ones are the ones that are sitting at other sites of the chromosome, preferably present in both libraries, like for instance this one. I would love to say that figuring out how this one is regulating capsule is as easy as it looks. Unfortunately, genetics rarely is. No, we are not just waiting for positive review in science, not yet. It's a bit more complicated than that. So right now, what we're trying to do is figuring out which ones are actually the true hits and weave them out from all the false positives, because we do see a lot of them. So I don't know exactly where this is going yet. But it certainly is exciting, and we're just still waiting for the next set of results to come in. So maybe, Kim, we can do this in another year's time, and you get the end, you get the second part of this story. But now, let's just say it's much more complicated than we initially thought it was. I think with this is usually the time where I would put in a lot of conclusions. I didn't do that today because I thought that if you've forgotten everything I ever said now, I just want you to remember one thing when you leave this room. So I put that here. And that is basically that the pneumococcus capsule structure, it is absolutely critical for its ability to infect its hosts, humans. And it is a very dynamic thing where it rapidly needs to build it and degrade it again, depending on the different niches, the different sites in the body where it is locating at this particular moment. I think there are a lot of near future experiments. There's a lot of obvious things that we need to do. And of course, these are the experiments that we are doing as soon as we are done here for the rest of the afternoon. I will leave you with a few bit more open questions, actually, that we want to figure out some point down the road. And that is, we know now, I think, why competence is induced. It's induced such that the cells can shed their capsule. We don't know how they do it, how it's a signal they send CSP, right? But how does that start? It's something the cells are producing themselves. So how is competence triggered in the first place? This is something that I think is the next big exciting thing. And of course, with this, can we inhibit this competence? Of course, as you saw, that the ability to shed the capsule is critical for this initial colonization, which is a prerequisite for further infection. So inhibiting competence, that could be novel drug targets for inhibiting the onset of disease altogether. And finally, of course, what we will do in a year's time, how exactly is the capsule biosynthesis, how is that regulated? Which genes are responsible for this? Finally, I'd just like to thank the people who actually did the work. That is PhD student Patrick and master students Jakob and Mikkel. And with this, I'll just thank you for your attention and take any questions.
just from the front row here, all the microbiologists. I guess you'll just...
I'll start.
Yeah, go!
Do you think that the composition is important as DNA transfer, or is it just a byproduct of breaking down the cell wall and releasing the capsule?
Competence is important. So there's a lot of things that I actually didn't have the time to talk about. It has just been published now. So in the nasopharynx, where the pneumonia cells are sitting, right, they're in fierce competition in that area with other bacteria, in particular Haemophilus influenzae and Neisseria meningitis. So they are constantly battling for territory. And what we just know now is that when competence is induced, there's also a set of proteins that is triggered that is killing off its competitors. And the streptococci is then just stealing its DNA and using it. So it's not just something that's a byproduct because of capsule shedding. It's also something that they actively use.
So you said there was a lot of false positives.
Yes.
Do you imagine if you include a third and a fourth and a sixth library of mutants, you would home in
On the right ones?
On the right ones.
Yes.
Okay.
That's actually a possibility, yes. It's very tricky experiments, and exactly what's going on, okay, I can only guess, but for now, we don't know, but we do see a lot of false positives. And that is, of course, that even though there are some obvious candidates, that seems that if we then go and make single knockouts of these genes, then we should see that they have a capsule minus phenotype. We don't see that, at least not on the candidates that we have tested so far. But yes, making more, I guess, is a possibility. Yeah. Yeah. Yeah.
So you mentioned in the beginning that there are 98 different serotypes, capsule serotypes worldwide. Would it be possible and realistic to develop a 98-valent vaccine? And if so, could it eradicate Streptococcus altogether, since humans are the only hosts?
So I guess there are several questions there. So let's start with, is it possible to create a 98-valent vaccine? According to pharmaceutical industries and the World Health Organization, they say no, it's simply impossible to make a complete 98-valent vaccine. It's simply too expensive to do that. And it's also a lost cause because there are currently 98, but I think just last year there were 93. So there's just all the time more and more new serotypes coming along. I think that answers the second part of your question. I think it's a lost cause to eradicate them altogether by having a vaccine like this, because there will simply be new serotypes coming along the way.
Kind of related to this question, you were quoting carriage rates in adults and children
Yes.
for Streptococcus. Presumably, though, from developed countries where practically all children are vaccinated against the pneumococcus. Right?
Yeah.
So does that then imply one of these two things? Either they're colonized with a serotype that they weren't vaccinated against, or that the vaccination is effective against just simple carriage?
The vaccine is not effective against simple carriage. But when we look at these carriage rates, it doesn't really matter which country we look at. This has also been done in countries that are just now adopting the vaccine, like Pakistan, for instance. So there we have data from before and after. And we can see that, so first of all, of course, there is a difference in how virulent the strain, of course, are. The strains that is covered in the vaccine, they are, of course, also the ones that are circulating in the children in the Western countries, right? These are the strains that that is included in the vaccine. So many of them would probably not be as effective in other parts of the globe where other serotypes are dominating. But we do know that the disease of the different serotypes, the number of patients, that is also correlated with the carriage rate. So it's really difficult also to say which strain is actually more virulent. I guess that answered a bit more than I guess.
I also have a question actually. So
Yeah.
are there differences in, so apparently they can give rise to a lot of different diseases depending on where they end up in the body. So
Yeah.
are there, like with the different serotypes, do they have a better chance of ending up, let's say, causing meningitis, crossing the blood-brain barrier, or?
Definitely.
Yeah.
Definitely. So I think this is in particular true when we actually just look at the vaccines that we talk about, because what is covered in the vaccine, that is predominantly the serotypes that are giving rise to meningitis. So there are certain strains that are much more common to find in meningitis patients than in pneumonia patients, where it will be other serotypes that are causing that type of infection.
Can I, one more question then?
Sure.
If there are no others. So I was really struck by your fluorescent microscopy showing that roughly half of the cells actually were able to go into competence. Do they actively communicate to, so that once a cell reaches competence, it will inhibit the surrounding cells from acquiring competence, or what is the mechanism regulating? Maybe I missed it, but I was.
The simple answer is we have no idea.
Okay.
I guess that's the simple answer. They certainly do communicate. But how they do it in relation to competence, and whether they are, the competent cells are making sure that the cells that are non-competent would stay non-competent. I think it's a bit hard to answer. Also, this bistability, it's also a matter of concentration. If we just add in more and more CSP to the cells, eventually, and given time, they will all become competent. So I guess with that logic, it's not something that these cells are inhibiting the others to do. But during physiological conditions, I guess it's possible. But we don't know.
Any further questions? Otherwise, I think we should thank Mikkel for a very exciting talk.